Okay, so first of all, I would like to thank the organizers for giving me this opportunity to present this work. And I congratulate both Mustan sir and Deepak, and hope that they will keep us showing the light in the future also. So today I will talk about this periodic forcing of adsorbed polymer and DNA. So we all know that in cellular processes like DNA replication and RNA transcriptions, the enzymes like helicases and RNA polymerase apply a mechanical force on the DNA to unzip it. By applying a constant pulling force on the ends of the DNA, it was found that the double-stranded DNA unzips to two single strands if the pulling force exceeds a critical value. And this is known as an unzipping transition, and it's found out that this is a first order phase transition. So this has been done some 2 decades ago away, and after that many more extensive studies have been done on unzipping transitions. One such study was in directed self-avoiding walk model in 1+1 dimensions. In this particular model, the DNA strands are represented by directed self-avoiding walks on a 2-dimensional lattice. So the walks start from the origin at origin and go towards a preferred direction. So in our case, it's a Z direction. And then at the end of the, they cannot cross each other. And at the end monomers, we apply a pulling force at the end. So the directed nature of the model allows us to write the partition functions, write the recursion relations, and then which can be solved exactly using generating function techniques and exact transfer matrix technique. And so using this these two techniques, generating function techniques, it was found that the force temperature phase diagram is given by this particular expression, and it's a first order phase transitions. So in this particular model, the thermal melting where the force is 0 is given by this particular expression, and it's a continuous phase transition. So now the question arises, so what happens when the ends of the DNA are pulled by a periodic force? So the answer is the force extension isotherms show hysteresis depending on whatever is the frequency of the pulling force. So here is an experimental thing. So from the Harvard group, so they used these magnetic tweezers. So we have our DNA hairpin here, where one end of the DNA hairpin is anchored at the at some support, and other on a magnetic bead, and which can be pulled by applying a magnetic field. So what they found is, they increase the force gradually, and measure whatever is the distance between the two strands. So this as you increase the force, the extension increases, reaches some maximum, and then come back. So there are two different cycles of this unzipping and rezipping. So is shown here, and we can see that there is a hysteresis in this particular case. So after that there are many other studies of periodic forcing of other biomolecules. They also show hysteresis. So the first study is shown by Sanjay Kumar, the previous speaker, and his co-workers. So they used Langevin dynamic simulations on short chain lengths. So like here is their model, a short chain with 16 base pairs, and they apply a periodic forcing f of t at one end, keeping the other end fixed. What they found is the DNA can be taken from a zipped state to an unzipped state, or vice versa, by changing the frequency of the force, keeping the force amplitude constant. So the area of the hysteresis curve, which serves as the dynamical order parameter, it scales as 1 by omega at high frequencies, and g to power alpha omega to power beta, or g minus gc to power alpha omega to power beta at a low frequencies. So their study showed that the exponents alpha equal to half and beta equal to half, which they later modified to alpha equal to 1/3 and beta equal to half. As we all know that the Langevin dynamic simulations are computationally very expensive. So we cannot go for very larger chain lengths. So we decided to check if these exponents are true by simulating longer chain lengths. And we used Monte Carlo simulations with 512 base pairs with very long chain lengths in this case. And thanks Rajesh for giving a very nice introduction to Monte Carlo simulations in the previous session. So then we use these two particular models. So one model is the same homopolymer DNA model, which I just explained earlier, for which exact solution is known. So in this case, now instead of a constant pulling force, we are applying a time dependent force, which is given by G of mod sine omega t, where G is force amplitude and omega is the angular frequency. And whenever the two strands, there is a contact, there is a contact energy epsilon in this particular case. We also study a heteropolymer hetero-DNA in this case, where the heterogeneity is given in the form of a blocks. So we have a block copolymer DNA of the type this AN, BN or BN, AN, where A type bonds has 2 hydrogen bonds, therefore having -2 epsilon energy, and B type bonds has 3 hydrogen bonds. So therefore we have this -3 epsilon energy. So now in this case, what happens? We anchored a force at anchored the force from one end and apply a force on the other end. So there can be two situations where you can anchor a B type base pair and apply force on the A type things, which is shown here for n equal to 4 case, or other way around also. So it so happens that this model also can be solved analytically. Exactly, it has an exact solution of the same expression which was given by these things, but now the epsilon will be given by the average of the binding energy between a block. But when you apply a periodic force, the result depends on which type of blocks the forcing is acting. So let me first talk about this homopolymer DNA model. So we use Metropolis algorithm, and then in this case, the extension xt follows the force with a lag. So here we have the 3 different cycles of the force and the extension at some frequency omega star. And so if we obtain the force extension isotherms by averaging over different cycles, we get a hysteresis curve. So these are the hysteresis shown at different force amplitudes for different frequencies, which are labeled here. And this omega star is the frequency at which the loop area is the maximum. So this loop area, so you can see that if you decrease the frequency, so the loop area increases, reaches a maximum and goes down. But when you have a higher force amplitude, you also see some secondary peak structures. So these are the 2 loops at omega star. So we could do a simple analysis to find out whatever is this omega star, the dependence of this omega star on the chain lengths from this particular expression. So just to mention that the stationary states for the 2 cases, higher force amplitude and lower force amplitude are different, and these oscillations what we see for higher force amplitude is because of these different stationary states.
Professor Kapri, you have 2 more minutes.
Yeah. So now we can see the secondary peaks at higher force amplitude gives the secondary structures. So we can find out the length dependent frequencies of these maxima and minima are given by this particular thing. So when the force area is plotted as a function of for different chain lengths as a function of omega n. We get a very nice collapse. From this thing, we can find out again the same, that A loop goes as 1 by omega at high frequencies. And so if you plot at low frequencies, so low frequencies here we have plotted here for both the scaling forms, omega g to power alpha, omega to power beta, or g minus gc to power alpha, omega to power beta. So what we get is the exponents alpha equal to 1 and beta equal to 5 by 4. So for the block copolymer DNA problem, so again we have shown the results for the low frequency regime here for 3 different sequences. So we again see that the exponents are your alpha equal to 1, beta equal to 1. So now from 2 different problems, we got the same exponent. So we thought that let me simulate a longer chain homopolymer DNA using LD simulations to check whether dynamics has anything to do. So here we simulated a bead spring model in 2 dimensions. So here is one of the snapshots taken from the simulations. Now the chain lengths are with 64 base pairs, an equal number of 128 monomers. So the trajectories are obtained using the LAMP software. So these are computationally very expensive. As I told, it took us 2 months to get these particular results. So again, we found out that this A loop goes as g minus gc to power alpha, omega to power beta, and the exponents what we got again is 1 and 1.25. So recently we have also studied the unzipping of an adsorbed polymer on a surface by a periodic force. So here we have taken 3 different type of surfaces which can be penetrable, both sides of the surface. There is same affinity to the polymer, penetrable, but polymer has different affinities on either sides or impenetrable. So the exponents what we get is again the same in this particular case. So the take home message from my talk is, so the DNA can be taken from a zipped state to an unzipped state by an oscillating force by changing its frequency, keeping the amplitude constant, or vice versa. The area of the hysteresis loop scales as A loop goes as 1 by omega at high frequencies, and g to power alpha, omega to power beta, or g minus gc to power alpha, irrespective of this scale is formed, but with the exponents are all alpha equal to 1 and beta equal to 5 by 4. So Suman Kalyan was a postdoc, and Ramu is a PhD student. Abhishek is my collaborator here. So we have discussed this problem at many times with Deepak and Suman Bhattacharjee. Thank you very much.
Thank you, sir, for such a wonderful talk. Any questions? You can raise hands or just unmute yourself and ask the questions. Yeah, there is one question in the chat box by Fazil Nazib. He's asking why the oscillating force.
So why the oscillating force? So normally, so if you see here, so there are experiments where normally, see in biology, normally these biological processes, you have things in a cyclic manner. So you these enzymes, helicases, they apply a force on the DNA, but normally what they do is they just use these ATPs and other things. So the force in a biological manner, biological problems are normally oscillating forces or something. So here we have taken a different sine omega t, but it may not be a sine omega t in that particular case, but it is a cyclic manner. They do have that part. So they apply in one so it's a basically a force is a cyclic order in this particular case. So that's why we have taken these things.
Any other question? Since there are no other questions, so let's thank Professor Rajiv Kapri for such a wonderful talk. Thank you, sir.
Thank you.